My name is Brandon, and I'm going over Lawam's work for the DNA and RNA page. Let's see. The notes I have for the DNA does a good job describing what DNA is, what the structure, along with talking about the building blocks and the base pairing. I enjoyed reading about what the DNA damage can do. I like how you inputted what it could do, such as brain diseases, aging disorders, immune systems, heart diseases, and birth defects, along with good visuals. I enjoyed the visual about base pairing and hydrogen bonds, talking about the nitrogenous bases. The methods were very clean, so they're easy to read and good design with a good flow. There's a good visual for the DNA purification. I like that with the flow chart. One thing is maybe not having one methods in between the two result sections that you had with the agarose gel. Also maybe discuss what A260 and A280 are with the ratio and what that means and why we use it. Also with the TAE buffer, why we use that buffer and what it means. And for this one, I thought it was really good discussion, talking about the future experiments. I think it did a good job describing what the data means and why we can apply it. I did think this page is overall very nice. And for the RNA page, again, is similar to the DNA page to describe what the RNA is, with the structure, the ribose sugars, the nitrogenous bases, and how they differ from DNA. That was good. Also talking about the different kinds of RNA and their functions, such as mRNA, tRNA, and rRNA, and just their role in gene regulation. It was a good job describing the P53, P21, the BAX, and the GAPDH as a housekeeping gene. I enjoyed the lengthy review on those for a gene overview, why we use them, what they do, and what their results would be in qPCR. I'd say maybe a little more definition on qPCR, possibly a visual can be added for that, but I thought the section was very good. The methods on that is a good flow chart. Talking about the cells we use, the lysis, the washes, the binds, and how we elute them. That was good. Maybe also, like before, discuss what the buffer RW1 is and RPE would be, why we use those, what they're used, what they do. Maybe got how the methods are very good and with their flow and organization. Also the subheader as well, describing the volumes for them. I thought the results section was also very good here. Talk about the nanodrop, the UV plus would be negative, each one for that, calculations that you did as well, describing that. I also enjoyed how you put the Bio-Rad I script in there. With that kit, that was nice to read. With the volumes and why we used that and how we did our experiment, that was nice. The QPUs, our results were very well defined. You're talking about cyber green, maybe talk about that a little bit more, but that was very nice. The nucleus-free water, our dilutants, that was good. And maybe just talking a little more with the delta CT too. I should do this well, but that was something, maybe a little more on that. But the bar graphs are very well inputted, with each figure having their own subtitle. And again, this discussion was good. I think that was it. The Western blots, RT-qPCR, talking about the reverse transcriptionase, which is maybe having a figure about reverse transcriptionase would be nice. Otherwise, this was very good. Thank you.